Today we'll be beginning with chapter 6 from the text and syllabus, which is titled Plant Nutrition. Now, the chapter begins with talking about photosynthesis. I'm going to come back to that later. And I'm actually beginning with the description of the structure of the leaf. So let's start with that. So what we'll be talking about mostly is a dicotyledonous leaf. Right, which basically means broadleaves. So generally, leaves are categorized as monocotyledonous or dicotyledonous, right, or either broadleaves or narrow leaves. So broadleaves are the ones that you normally see around you, and narrow leaves are the ones that, for example, if you've seen the leaves of a wheat plant or grass, so they're long and narrow. So those are monocots, right? Dicots and monocots for short. Now, whatever leaf it may be, right? It has a stalk that joins it to the stem of the plant. So there's a leaf stalk. That attaches the leaf to the stem of the plant. And right when the leaf stalk goes into the leaf, it's called the midrib. So, right, a leaf, right, and then this thing. Outside, this is the stalk. Inside, this is the midrib. Right, and from the midrib, you can see a network of branches coming out and spreading out throughout the leaf. Right, sort of like a capillary network, although we haven't formally talked about that. You may be familiar from your earlier classes. So, right, those are veins. So it's a network of veins. Right. So to repeat, a leaf stalk is, right, what attaches the leaf to the stem. So, right, this part over here, right, and let's say this is the stem of the plant. This is the stem, plant stem. So you got a network of veins, and we'll see when we look at the structure that these network of veins are responsible for a few things, right? They supply water and nutrients to the leaves. They transport, the products of photosynthesis, which again we'll come back to later. From the leaf to the rest of the plant. And they also provide structural support. So they're sort of like the skeleton of the leaf, right? And so they're spread throughout the leaf, and that helps the leaf maintain its shape and structure. Because inside the leaf, as we'll just see, right, it's all soft tissue. Now, the leaf is adapted to its function of photosynthesis because, to just briefly mention, that in photosynthesis, in the leaf, there is an exchange of gases, right? Oxygen and carbon dioxide, uh, and absorption of light from the sun. And so, how is the leaf adapted for photosynthesis? So, for one thing, generally they have a broad, flat shape. They are thin. So let's talk about the broad, flat shape first. What this does is it provides a large surface area for gas exchange. That makes the gas exchange quick and efficient. Right? And it also gives you a large surface area for absorption of sunlight. The next is that the leaves are thin. and so they reduce the distance required, for the gases to diffuse. When we discuss diffusion uh, back in transport uh, across membranes, I believe, or whatever the formal title of the chapter is, movement into and out of cells. Right, that's chapter 3. And one of the features that affects the speed of diffusion is the distance across which particles need to diffuse. So, right, the leaves are thin, and that reduces the diffusion distance by quite a lot, obviously. And so carbon dioxide is easily able to diffuse in, and right, it also makes it effective for sunlight to penetrate the leaf and get to the cells that contain the chloroplast. Now let's talk about the parts of the leaf in a bit more detail, right? So what are they, and right, what is their role? Now for this, first I will take you to an illustration in the text that will just make things easier, as opposed to me trying to draw it all out. Put that on screen. All right, so this is a leaf, obviously, and you can see the leaf stalk, the midrib, sorry, the veins, the network, and the blade or lamina, which is basically, right, the surface of the leaf. And then this is a transverse section, so this is the leaf is cut across, right? Its width, not across its length. If it were cut across the length, then it would be a longitudinal section. This is cut across its width, and so that's a transverse section. And what you can see is the midrib and right the vein, and then inside the midrib, right, this part over here, they are magnifying it, and so you can see this complex structure of cells. So I can zoom in over here and take a better look at what we have. So the midrib is this part, right? This whole thing. And then you've got the other leaves of uh, the other cells of the leaf. And over here something similar to the midrib. Right, obviously it's coming from the one of the veins that are branching off from the midrib. You can see similar structures. Now what do we have? The first thing we have, you can notice, is a very thin layer. You can just barely make out a line going over the top. That is called the cuticle. Then you have this first layer of cells. It's a single layer of cells going across the top. That is called the upper epidermis. Then you've got these long column-like cells, right? Have them going pretty much all the way through except for the midrib. So they're not in the midrib, right? But then throughout the lamina of the leaf. And those are called the palisade mesophyll cells. Below them, you've got these sort of random shaped and spaced out cells, right? These are called the spongy mesophyll cells. Then, right, so they're spaced out, there's space between them, and those are obviously air spaces, so they have their function. Then you've got this layer of cells at the bottom, and that is the lower epidermis. And in the lower epidermis, you've got what are called guard cells. So you can see over here, right? Over here, if you notice, you've got these two semicircular cells. So that's not one whole thing. This semicircle and this other semicircle, right? These are two guard cells, right? Only one has been labeled, and then this black hole in between the two of them is called the stoma. Stoma is singular. What we normally hear is stomata, right? So that is an opening giving access for gas exchange into and out of the leaf. Right, so the vein and then the midrib both have what are called xylem vessels and phloem vessels. Right, over here as well you can see xylem, which are more on the inside, as you can see right from the labeling. They're more on the inside, and the phloem sieve tube or the phloem vessel, they are on the outside of, whether it's the midrib or the stem of the plant, which we'll come to next chapter, or the root, because these two vessels travel throughout the leaf. But again, not to confuse, right? So let's throughout the plant. Sorry, I seem to be making a few mistakes and I'm slower because, right, I'm not feeling my best, so apologies for that. So the phloem sieve tube, and they have the function, the xylem vessel brings in water and nutrients from the soil, and the phloem sieve tube takes away the glucose that is produced by photosynthesis. Right? Now, if you can just sort of retain this structure in mind, we'll talk about what these different structures do, and also note, right, you can see these green dots in the palisade mesophyll and the spongy mesophyll. So those are chloroplasts, right? Chloroplasts contain chlorophyll. We've talked about these back in chapter 1. And right, this is where photosynthesis takes place. Any questions about the structure of the leaf? What is the spongy mesophyll does, take part, I mean, it does carry out photosynthesis, but primarily that happens in the palisade. The spongy mesophyll is more responsible for the movement of water through the leaf and also the gases, the diffusion of gases, right? It does contain chloroplasts, so right, some level of photosynthesis does occur. Let me just go back to the whiteboard. So parts of the leaf and their function. So the first thing we have is the cuticle. And this is a thin waxy layer, right, on the upper and lower. So it's the cuticle is actually present on both sides of the leaf, right? It's not only on the upper side. So a thin waxy layer on the upper and lower surfaces of the leaf. And right, what this does is it reduces primarily loss of water. So it's a protective barrier. Uh, I wouldn't use that analogy. Uh, you could, I mean, I maybe I'd call it the cell wall of the leaf, but it's not a cell, obviously. We're obviously trying to compare and understand. It's just a protective barrier for dust and microorganisms as well that prevents microorganisms from entering the leaf for like it's like the skin. I think using the analogy of skin is better. It's like the skin of the leaf. And right, it helps reduce water loss. Right, so again, protective barrier for example, from microorganisms, microorganisms that would be harmful to the leaf. Then you got the layer of the upper epidermis, and right, those are like thin, tightly fitting cells, and they are transparent to allow light to pass through. And again, they also act as a barrier to disease organisms. And these are the ones that actually secrete the waxy substance that makes up the cuticle. Just a sec. Secrete means that they make something and then release it outside the cell. All right? Does that make sense? Next up, we have the layer of the palisade and spongy mesophyll cells. So the mesophyll tissue. And this obviously contains of the palisade and spongy mesophils. The palisade coming first, and then the spongy. So the palisade cells are long and they contain a lot of chloroplasts. They contain many chloroplasts, and they are the primary site of photosynthesis. Right, so you know what chloroplasts are. They contain chlorophyll, which is a green colored pigment, right? Then we have the spongy cells, so spongy mesophyll cells, or the spongy tissue. They are irregularly shaped, and right, so the palisade cells, they are, as you've seen, regular shaped, and they are packed together. Packed close together. The spongy mesophyll cells don't have any regular shape and are loosely packed with air spaces in between them. Right, and they also contain chloroplasts. So, right, obviously, as you mentioned, these are the site of photosynthesis, and the maximum photosynthesis occurs during the day, right, when there is sunlight, and it reduces to almost none during the night. And so during the day, you've got carbon dioxide coming in, oxygen being removed, and during the night, you've got oxygen being taken in, carbon dioxide removed. And that happens through the stomata, which we'll just talk about. So talking about the air spaces, do we have space here? No. So the air spaces, right, are important and used for the diffusion of gases and for the loss of water vapor via transpiration. So that's something we'll talk about in the next chapter, transpiration. So, right, water vapor evaporates from the surface of cells. It becomes, and then, is removed from the stomata. Then we have the guard cell and the stomata. Right, so the guard cells are usually present or right in as pairs, right? They are semicircular in shape, and right, the opening between them is called the stoma, which is singular, right? And the plural is what, as I mentioned, what we regularly usually hear or use, stomata. Now, in dicotyledonous plants, the ones with broad leaves, stomata are present only in the lower epidermis. So again, just as a reminder, broad leaves, only in the lower epidermis, whereas in monocots, they are equally distributed in the upper and lower epidermis. Okay, so we've talked about osmosis in plant cells and how they become turgid, right? And the effect, like the role of the cell wall in maintaining the shape of the the plant cells when they become turgid. So in the stomata, well, not stomata, sorry, the guard cells, that is, you know, the concept that's at work. So what happens is the guard cells are like this, similarly. So those are individually guard cells, and what I'm going to do is I'm going to draw black over here on the inside to show that this part of the cell wall is thick. So the cell wall of guard cells is thicker on the side that is surrounding the stoma, right? And so when these cells become turgid, they increase in size. Now, what happens because of this thicker, uh, you know, cell wall is when the water is pushing, right? Which part of the cell wall do you think will expand more? The one that is, the regular cell wall, or the one that is thicker? Yes, Haram?
The regular one.
The regular one will expand more, right?
Yeah.
And so, right, as the cell grows, grows in size, it increases in size, it pulls, right, the, you know, uh, the stoma open, because, right, there's more expansion of the cell happening over here. The cell is not increasing over here, it's not increasing in size. And so, right, it's pulling this part as well as it's increasing. So the stoma opens. On the flip side, right, when at night, so this is happening during the day. At night, the guard cells lose water, right, and they're no longer turgid, so they're not, you know, they're not in an expanded state, and so the stoma closes. Does that make sense?
Yeah.
Okay. So obviously, right, when the stoma is open, you have, carbon dioxide can easily pass in, but water is also lost by transpiration. Whereas, when the stomacher is closed, right, CO2 cannot be absorbed, so that affects photosynthesis, and water is also not lost. So the opening and the closing of the stomata is a very, it's a delicate balance for the plant, and it's tricky. I mean, for us it's tricky. The plant is doing just fine. It knows what to do. It knows how to survive. And so when it's hot outside, or when it's windy, or when it's humid, or maybe when there's too much water or too little water, and so how does the plant manage, right? If it wants to reduce water loss, If it's hot outside, it will close the stomata, but then that will also decrease photosynthesis because you're not getting carbon dioxide that's coming in, right? And if you want to increase the rate of carbon dioxide, then you open the stomata, but you also risk the loss of water, right? So that there's a balance to be maintained by the plant in that regard. Okay. So how does the opening and the closing actually happen, right? Why does osmosis, like why is water entering the stomata? So there's a theory, and there is a better understanding of this. Obviously, if you choose to take biology in A level, you'll learn about it in more detail. But what they mention over here is that potassium, right, a mineral, and so in the cell it's present as potassium ions. And so how does that happen? So potassium concentration in the guard cells, right, in the vacuoles of the guard cells increases. And so what that does is that lowers the osmotic potential, the water potential, right, of the cell sap. And so water enters the guard cell. Okay. Uh, talked about the mesophyll, talked about the air spaces, and just quickly to wrap this up, we are going to talk about the vascular bundles, the veins. Which number do I put here? 6. And they contain the xylem vessels and phloem tubes or phloem sieve tubes. So the xylem vessels are long and thin, and the cells do not have any contents except for the cell wall. So there's no nucleus, there's no cytoplasm, none of the organelles. And so what that does is that just you have these cells that are joined together, right? And the only thing left of them is their cell wall on the outside, and that gives you a clear, constant tube for water to move through. And right, this happens when the cells are mature, and they also secrete a substance, right? They're lined, their cell walls are thickened with a substance that is called lignin. And so they carry water and salts from the roots all the way up to the leaves. Now, next up you have the phloem, uh, right? And that is made of sieve tubes, so those are living cells. And right, the cell walls have holes in them to allow the passage of materials. And the process of the passage of the material is called translocation. And these holes are called sieve plates, right? So they're obviously different from what the xylem is. And so photosynthesis requires water as well, and so that is provided by the xylem. And so from the xylem, right, it brings in water and it moves through the mesophyll cells, right, via osmosis. From cell to cell, it spreads out depending on where it needs to go. And the sugar that is produced during photosynthesis, right, that diffuses and goes to the phloem. Okay. Uh, right. I believe that is all that there needs to be said about the structure of the leaf. Just one thing. The guard cells are the only cells of the lower epidermis, and this is for dicots, obviously. For monocots, they're on the upper epidermis as well, that contain chloroplasts. Now, chloroplasts are not only found in leaves. If you have a green plant, if the stem is green, right, then that green color is coming from chloroplast. That means that the stem of this plant also contains, the cells of the stem of the plant also contain chloroplast. So the leaf stalk, if that's green, then the leaf stalk also, the cells of the leaf stalk also contain chloroplasts. But just having chloroplast doesn't mean that those are sites of active photosynthesis, right? So why do these parts of the plant have chloroplasts? That is, right, perhaps not well understood and definitely beyond the scope of the syllabus, right? But just to mention that. So because questions like this have come in the paper to identify which cell in the lower epidermis has chloroplast, so guard cells, right? They are the only cells in the lower epidermis that contain chloroplast. And that brings me to the end of today's class. Do you have any questions?
No, like, but spongy mesons also have chloroplast, no?
Yes. But the spongy mesophyll are not the lower epidermis.
Oh, okay.
Okay.
They are in the upper epidermis.
No. The upper epidermis is a single layer of cells. The lower epidermis is a single layer of cells. Everything in between is the, you can say, the cell tissue. Although upper and lower epidermis are also included in that, but the mesophyll layer is separate. So you've got the upper epidermis, which is one layer, the lower epidermis, which is the other layer, and then you've got the mesophyll layer, in which the palisade mesophyll comes first, and then the spongy mesophyll comes next. So they're not part of the upper or the lower epidermis; they come in between. All right.
Yeah.
Anything else?
No.
All right. So we'll continue this next time, inshallah, and
Can
have a good
you
week.
send
Huh?
can you please send these notes like after the class?
Sure.
Thank you.
Okay.
Allah Hafiz.
Allah.